talk about ribosomes and the structure and function. So ribosomes are organelles that combine the functions of mRNA and tRNA to actually make polypeptides. So in this first diagram, we just see the basic shape of a ribosome, which has nothing bonded to it, or the sites are not in use. So there are two subunits, the larger one and the smaller one, and there are two tRNA binding sites, which are right here, and an mRNA binding site, which is right here. And each of the subunits are made up of protein and a special kind of RNA called ribosomal RNA. And then down here, we have the same ribosome when the binding sites are actually in use. So right here, we have the mRNA that is bound, and we have the tRNA and the codons, and we can see that the tRNA is held close to the two binding sites are actually close together so that the amino acids carried by the tRNA molecules connect into a polypeptide chain, which is here. And then here's the next amino acid that's going to be added to the polypeptide chain. So in this slide, we're going to be talking about how translation begins, which is initiation, which can be broken up into two steps. And the first step is what is pictured here in this picture. So an mRNA molecule, which is right here, is binding to the small ribosomal subunit, and then the special initiator tRNA molecule is binding to the specific codon called the start codon. And the start codon is on a place on the mRNA strand where the translation should begin. And the initiator tRNA has the amino acid MET, which I didn't write in here, but that's what it is. And its anticodon UAC, which would be these three spikes right here. And to the start codon, which is AUG, the opposite, and you know that from the specific bonding rules. And then in step 2, the large ribosomal subunit bonds to the small one, creating a functional ribosome. So I'm just going to draw that in right now. So the large ribosomal subunit is bonding, and now we have a functional ribosome. And so the initiator tRNA fits into one of the binding sites. So then we have this other one right in here that is not occupied. And this site is the P site, and it has the growing polypeptide. And then this is the P site right here, labeled with a P. And this is the A site right here, which is vacant, but is ready for the next amino acid bearing tRNA. So in this slide, we're going to talk about how amino acids are added to a growing polypeptide chain. So there's a 3-step elongation process that occurs. And the first step is codon recognition, the second step is peptide bond formation, and the third step is translocation. And then to stop the process of addition of amino acids to the peptide chain, we introduce the stop codon, which acts as a signal to stop translation instead of encoding for more amino acid production. So over here we have a diagram to show. This is actually a cycle, but I just didn't draw it out that way. So in the first ribosome, we have the P site occupied by tRNA, and then the A site is open for another to come in. And over here we see the polypeptide chain, and then here's the other amino acid that will eventually be added, and these are the codons on the mRNA. So then here, the codon is the anticodon, which is right here, of the other tRNA molecule pairs with the mRNA codon in the A site, so that would be right in there. And then in the second step, it is peptide bond formation. So the polypeptide, which was attached to the tRNA, attaches by a peptide bond to the amino acid in the A site, so it's going to attach on to this amino acid right here. And that attaches by a peptide bond, and the ribosome catalyzes the formation of the bond, so then another amino acid is added to the chain. And if you look back at the previous slide, a couple of slides ago, we actually talked about why the ribosome catalyzes this bonding. So in the third step, which is translocation, the P site tRNA leaves the ribosome, and then the ribosome moves the tRNA in the A site with the polypeptide chain, which has one amino acid added from the previous step, which is right over here, to the P site, and then the codon and the anticodon are bonded, and the mRNA and tRNA move as a unit. So this movement brings into the A site the next mRNA codon to be translated, and so then the process begins again as we cycle back, and then the stop codon reaches the ribosome site and is the termination, and it acts as a signal to stop translation. Alright, so this slide is basically just going to be like a summarization of everything we've talked about. And so we know that the flow of genetic information in a cell is from DNA to RNA to protein, and in our steps of transcription and translation we accomplish that. So in this is occurring in the nucleus, and it is demonstrating transcription. So here's the DNA, the mRNA, and the RNA polymers, and the mRNA is being transcribed from the DNA template, which is when the DNA strands separate, and the mRNA, the RNA is synthesized, and it occurs in the nucleus, like I just said, because the mRNA has to travel from the nucleus to the cytoplasm, which is where translation occurs. So then in translation we have this amino acid, and we have an enzyme, and we have a tRNA, and some ATP, and that's going to, the enzyme, which is really specific to the amino acid, is going to attach these two together to form, and here we have the anticodon, and then here we have our ribosome with the large and small subunit, and the initiator tRNA, and the